So we've learned a lot about DNA and the structure of DNA and how DNA is used to make proteins and protein synthesis. And this is just kind of a recap of that bigger picture, how important the genetic code is to all living organisms. So you can think of DNA in the genetic code as being like a language, and it's a way to relate these nucleotides in mRNA and DNA to amino acids, to these protein products that do things to sustain life in organisms. As we know, the order of the bases corresponds to specific amino acids, and the order of those amino acids dictate the folding pattern of our proteins, which dictate the function of our proteins. One thing that's really astounding to me is that the genetic code and DNA and RNA is so universal. It's in almost all organisms. They all use the same sequence of 3 base codes for the same amino acids. So all living things have this in common. We all use the same basic genetic code and language in life. So there are 64 possible triplet combinations from the 4 bases A, G, C, and U. Since there's only 20 amino acids, that means that one amino acid can have several codons. And glycine, the amino acid glycine, is a good example of that because GGG, GGA, GGC, and GGU all code for the amino acid glycine. UGA, UAA, and UAG are stop codons. And when you're reading your codon chart, that's not something you're going to have to memorize, but you will see the word stop, and that's okay if you are translating that into amino acids on the paper. If you come across a stop codon, you should put stop. You should not put any other amino acids in that position. AUG is our only start codon. AUG is a start codon, but it also goes by methionine, so you may see that used interchangeably. All right. So let's do a practice question that you may see on a quiz or a test. So for example, I would give you the DNA. If there are not 2 strands, you can assume that that's the template strand. That's the strand I want you to read and create an mRNA transcription from. So to do this, we need to remember that A in RNA pairs with U and U with A, and G pairs with C to make our mRNA transcription. So T pairs with A with U, C with G, and I'm on to my next codon. And I personally like to write lines between them or count every 3. That helps me to track it with my eyes. Totally your call on how you want to do that. The next set, C, C, G, then G, G, A, and then U, G, A. So now that I have my mRNA transcription, I can go to my codon table and I can start to figure out what the sequence of amino acids is based on the mRNA, based on the DNA that it came from. All right, so AUG, AUG is methionine. This is also known as our start codon. So methionine, our start codon, is the first, and you should see that as the first. If for whatever reason it's not on your test and you're sure that you did it right, it's okay. That's more of a formality. That's how it happens in real life, but sometimes I'm just giving you letters to try to use your codon table. All right, the next one is CCG. So there's C, C, G. So I went down that column, and that's proline. You don't have to know the actual names. If your codon table does not give you the full name of it, using that 3-letter code is just fine. GGA, so there's G, here's the other G. GGA is glycine. We saw that on the last slide. And UGA is a stop codon. Again, notice that this actually says stop. It doesn't say anything else. That's appropriate to put on your quiz or exam as being a stop codon. So this DNA sequence codes for the protein with the amino acid order methionine, proline, glycine, stop.